Hello, class. I hope all is well. So this is where we stopped last time, the homeostasis. The definition of homeostasis is that you should know, so don't say Amir, it was not in a PowerPoint. The definition of homeostasis is that the internal condition of the body of an organism, you're taking zoology, of an organism must remain constant, okay? Let me turn on the light so you guys can see. Okay, it's a little bit better. You can see my face now. So the internal condition of the environment must remain constant within a tolerable limit. Okay, so there is a limit. For example, let's say your body temperature. If your body temperature goes above 37 Celsius, 98 Fahrenheit, then you have fever. If you have body temperature of 39, 40, 41, God forbid, you have fever, okay? So, but when you go within tolerable limits, 39, you probably don't go to a doctor, 38, 39, you probably stay home, do not do anything. But when you get to 41, then you should go to a doctor. Okay, so that's the upper level. The bottom level, if your body temperature falls below 35, 34, then you're hypothermic. You got Houston, we got problem. Okay, so within tolerable limit, your body can tolerate some limit. And that's what homeostasis is. So your sugar level in your blood goes up and down. We already talked about calcium level in your blood goes up and down. So that's about most everything in your body. It has to be within tolerable limits. So based on your textbook, circulatory system, respiratory system, urinary system, endocrine system, and digestive system, and immune system, they all work together to keep the homeostasis. But, you know, I put a few dots at the end right here. I would like to say it's a whole organism. It's an entire body. It's just not, your body is not just those systems. Everything works together. And that's, of course, the next few chapters we're going to discuss and talk about. But, you know, that's what I feel like. It's a whole body. Your homeostasis is your whole body. Okay. So let's talk about extracellular fluid, the fluid which is outside of the, there are two types of them. Let's see, fluid further divided into two, blood plasma and interstitial fluid, or intercellular fluid, tissue fluid formed from plasma. So if I grab your blood right here, so if I grab your blood and put it in the test tube, and what happens after a while, or if I spin it, right here, that would be your red blood cells. And there is a band on top, they call it formed elements. That would be your white blood cells and platelets. And right here, up to here, that would be plasma. Oh, I should have dropped the arrow to that one. Plasma. Okay. So the plasma is the fluid outside of the red blood cells, and that is called a blood plasma, intracellular fluid, or intercellular, interstitial fluid, or intercellular fluid. And then we have another fluid which is inside of the cell. You took biology 1. Inside of the cell, there is a fluid plasma. Cytoplasm. Cytoplasm, that fluid is called intracellular fluid. Okay, I'm going to write it down here. Intracellular fluid. Okay, so that is the fluid which is inside of the cell, and that is intracellular fluid. Let's see. Okay, so in here, oh, come on. Intracellular fluid inside of the cell, and then that would be all of the cells. Your white blood cells, they are broken down to 5 different types that you should know for this class. And you take hematology or other classes, you learn more. But there are these, I call these three, where are you? These three, I call them three musketeers. So they have granules, eosinophil, neutrophil, and basophil. Eosinophil, it picks up the acid when they stain them, it picks up the acid. Neutrophil does not pick up acid or base. And basophil, guess what it does pick up? It picks up the base. So when they prepare the slides, they put a very thin layer of blood on a slide, microscopic slide, and they pass them through different solutions, different acid, base, neutral, and so on and so forth. And these cells pick up the acid. If they pick up the acid, and then it's acidophil, another name for right here for eosinophil is acidophil. The name eosin is a type of the stain they use. It's called eosin. And basophil is, it picks up base. That's what they call it, basophil. Phil, as you know, it means love, like. Neutrophil, it does not pick up acid or base. So those are the granules inside of all of these. You can see these are granules inside of, that's a nucleus right here. You can see the nucleus. Maybe I should, oh, sorry. This is the nucleus right here, and this is the nucleus. And these granules, they're little packages, vesicles that they contain enzymes, and they contain, and you will see the next one, histamine and other things. These two cells, they do not have granules. Okay, they do not have granules. So they call them in textbooks, and here we call them agranulocytes. Okay, these are what? Granulocytes. Okay, so agranulocytes are two of them, monocytes and lymphocytes. Monocytes are 3 times of red blood cells. Lymphocytes are same size as red blood cells, and the nucleus covers most of the cell. On the microscope, you will see. And monocytes, there are 3 times of red blood cells, and nucleus is C-shaped. Do not go by shape of nucleus in eosinophil, neutrophil, and basophil. Okay, do not go by the granules. Okay, the granules are orange, red. They are acidophil. The granules do not pick up. You see the cytoplasm without any granules in there. That's neutrophil. You see the granules on the microscope. Do not go by nucleus. You see the granules on the microscope. They're blue, purplish, dark. That's basophil. And then, of course, you have platelets, which there is a scientific name for platelets, erythrocytes, or red blood cells, and leukocytes, or white blood cells, and platelets, or thrombocytes. Thrombocytes. Okay, that's what they are. Here is red blood cells, lymphocytes, same size as red blood cell. Okay, monocyte, 3 times of red blood cell. Nucleus is C-shaped. They do not have granules. Nucleus covers up most of the cell. Basophil, these are the models we have in the lab. 2.5 times of red blood cells. Eosinophil, 2.5 times of red blood cells. And neutrophil, 2.5 times of red blood cells. On the microscope, of course. The models here are a little bit out of wacko, but anyway, that's okay. It's a good model. Here they are, the functions of the each blood, and the percentages when you look at them, when you go to a doctor and they look at your blood. If any of them is out of, you do not have to memorize these percentages, but they look at these and they say, yes, you're within the range. No, you have parasitic infection. So here, right here, you have a lot of this. And you have parasitic infection right here. You have severe allergy. So you have more in here. But know that the least amount of white blood cells in your blood is basophil. It's very rare to find them on a microscope. Okay, it's very rare to find them on a microscope. But the rest, the most common one is neutrophil. So that you should know. The least one and the most common one are neutrophil and basophil is the least one. These are the functions that you should know. Phagocytes, small particles, neutrophil. When there is an infection in the body, they act like the policeman or the fireman. They arrive to the site of infection first. Okay, that's what they are. That's what neutrophils do. And then eosinophils, they kill parasites, which we study a lot of parasites in here, help control inflammation and allergic reactions. Basophil release heparin and histamine. That's why when you have, you start with sneezing with the pollen, if you ingest pollen, then you take antihistamine. These guys, basophils, are doing their job. They're trying to neutralize those pollens, but they release too much of them, and that's why you have allergic reactions. You take antihistamine drugs. Monocytes, phagocytes, large molecules. Neutrophil, phagocytes, small molecules. And then lymphocytes provide humoral immunity, which makes antibodies, which I will talk about it here in a minute. Oh, sorry. Let's go. Blood composition. Plasma is 90% dissolved solids, dissolved gases, formed elements. I talked about that. Red blood cells and white blood cells and cell fragments, thrombocytes. I talked about that. The proteins in the blood are albumin. You should know that. You're not taking physiology, but quickly you might someday. It will help you. I broke the chair. Sorry. So let's put it back so my wife doesn't complain. Albumin, osmotic equilibrium, and globulins, which you're going to talk about it next, is for immunoglobulins like IgG, IgM. You will see that acronym later on. And then fibrinogen for coagulations. If you cut your finger and start splitting, then fibrinogens are protein molecules. These are some of the protein molecules that are found in the blood. Okay, cells, let me see what it says. Cells of the acquired immune response in vertebrates. So lymphocytes, they are B lymphocytes, they make antibodies. T lymphocytes, they have surface receptors, and T cells, subsets CD4, CD8, clusters of, that's what CD stands for, cluster of differentiation. And the AIDS patients, they have, they take the monitors CD8-4 and CD8. Okay, so sorry about that. That was myself. Always I get text or message or something. T helper cells, and then TH1. TH stands for T helper cells and T helper cells, and natural killer cells and mast cells. So those are. Let me go back to natural killer cells and mast cells. They are the cells that they go around in the tissues and they just wipe out anything that is unwanted. And they engulf and eat them. That's what they call them, natural killer cells and mast cells. Okay, the next one, I will talk about how antibodies are produced, but this is the structure of an antibody. I would like you to know something about it. Antibodies, we have maybe I should talk about it now. Damage. Okay, so damage is IgD, IgA, IgM, IgA, I already repeated it, IgG, IgE. So there are 5 major different type of antibodies. And then the very first antibody that appears at the site of infection that is made by body is IgM. And IgG is later on during infection. IgG comes in, and that's what the doctors look for. If they want to know you're infected with something, let's say, corona, they look for IgG, or food poisoning, they look for IgG, or Toxoplasma gondii, they look for IgG. IgM is not very particular, it's not very specific, and I will talk about that, but IgG is more specific. So having said that, most antibodies look like Y, and there are 4 chains of protein molecules, 1, 2, 3, 4. Of course, number 2 and 3, it's a long chain. And number 1 is attached to number 2. It's called light chain, heavy chain. The 2 blue one is called the heavy chain. The light chain are attached to blue chains, this light chain, this one, by a disulfide bond right here. And in Bio 1, you studied disulfide bond, the amino acid cysteine, it has sulfur, and with another cysteine from another protein molecule, they can form a covalent bond, which is called disulfide bond, okay? That is in Bio 1. And right here is another disulfide bond, another disulfide bond combined the heavy chain to heavy chain. And then here's another light chain is connected to the heavy, this number 3 heavy chain by disulfide bond. So what happens, the amino acids, this area right here, if you would, that I draw a box around it, I draw a box around that, all of the amino acids in here, in here, in here, in here, in here are constant. Okay, so it doesn't matter if you're you've been infected with food poisoning, or you've been infected with coronavirus. It doesn't matter, or Toxoplasma gondii, or some worm, or something. The amino acid sequence in those regions that I showed you right here, right here, right here, right here, right here, are pretty much same. What is different is the amino acid, they call it in the variable region, the textbook called the variable region or antigenic region right here. These area, okay, they're showing it to here. That area right here and right here, antigenic binding site, okay, that is, the scientists are able to detect you've been infected with COVID, or you've been infected with food poisoning, or you've been infected with HIV, God forbid, and so on and so forth. Okay, I hope I'm making some sense. So the sequence of amino acids in this area are varied, they're different. Okay, how does your body make amino acids? Okay, the amino acids for COVID, the amino acids for food poisoning. I've been emphasizing on those right now so far. So let me talk about those two. What happens here is a viral, they call it viral antigen. It could be a bacteria, it could be a parasite, it could be anything. Your textbook from a long time ago, they wrote on viral antigen. Antigen is something that stimulates production of antibodies right here. These are antibodies, Y shape. You remember that? I told you it's Y shape. Okay. So right here is a viral antigen, and the name of this cell that they have receptors for that antigen, okay, is called antigen presenting macrophage. That's the name of that cell, antigen presenting macrophage. Antigen presenting macrophage, when recognize this virus, it releases a molecule called interleukin. I spell it out for you guys. Interleukin will stimulate the next cell, a T helper cell, and then T helper cell releases another interleukin, stimulates another T helper cells, and another T helper cells, and eventually they stimulate B cells. Now, B cells can differentiate to 2 different types of cells. They differentiate to plasma cells and differentiate to memory cells. Memory cells stays in your body for the next infection. They come in, and that's what vaccine is all about. They come in and they bypass most of this right here, and they go into work immediately. They turn into plasma cells and they start making antibodies. So in Bio 1, you study these. These are rough endoplasmic, rough ER. These are rough ER. They make protein molecules from Bio 1. And then the protein molecules we are talking about are amino acids. Those amino acids attach to this antigen, this virus. And then this cell, phagocytic macrophage, will engulf, with the presence of this virus, it will engulf the virus into the cell, and with the help of lysosome, again, in Bio 1, it will kill that virus. Okay, I hope it makes some sense. Without the antibody, okay, without these antibodies, the virus cannot be engulfed, cannot be eaten by phagocytic macrophage. That whole process that I explained is cellular humoral immune response. Is humoral immune response. At the end, this portion of it, it would be cellular immune response, but all the way to here is cellular immune response. The new picture of your textbook is this one. Same thing I talked about it. Here's your antigen, okay? And then it makes interleukin. Interleukin, it will stimulate both humoral immune response and cell-mediated immune response. Eventually, the antigen is going to be engulfed by the phagocytic macrophages. Okay. The next one is to talk about your blood, your heart, and the circulation of the blood in your heart. I should make something clear right now, because I had students one semester, at the end of the semester, they said, "Oh, really? We don't have a pump that pumps the blood into our heart?" I said, "No, you do not have a separate part in your body to pump the blood from your toe or from your brain into the heart. The muscular activities you do," okay, I cannot show my legs. When I move my head, all of the muscular activities I do, the veins in the body are embedded in the within deep in the muscles. So this is my vein. Imagine this is a vein, and these are skeletal muscles. When you move them, they push the blood into the heart. So we do not have a heart to push the blood into our push the blood from our head or from our toe into our heart, or our finger into our heart. We do not have that. It's your muscle contractions. Okay. So having said that, all of the blood from your head, from your toe, they come into the heart. It's a cycle, so it doesn't matter where I start. But most textbooks starts with right atrium. Let's start with that. So blood from upper portion of your body, anything above the heart is called from superior vena cava. Blood comes, it's a big vein that brings the deoxygenated blood, the blood that has been used. It gave off this oxygen and picked up carbon dioxide from tissue like muscles, liver, bones. They bring them into right atrium, right here. And then the blood from lower portion of your body, all the way from your toe, they come through inferior vena cava by muscular contraction. There is no vacuum in your body to suck up the blood from your toe into your heart. So all of the blood accumulates in the right atrium, deoxygenated blood. And then there is a valve between the right atrium and right ventricle, which is right here. And it's called right atrioventricular valve. I like that term. It's easy to memorize. What is the other one? Mitral valve and other things. You know, I don't like them because I forget them. Okay. So the blood goes from right atrium through this valve, right atrioventricular valve, to right ventricle. Right ventricle squeeze. Okay. When you squeeze, there is a valve right here that the blood does not flow back. That valve is called pulmonary semilunar valve. So the blood from this right ventricle squeeze, it goes through the right semilunar valve and it goes to pulmonary arteries. The only artery in the body that carries deoxygenated blood. The rest of the arteries in your body carry oxygenated blood, not this kind. And then the blood goes into your lungs. They get oxygenated And I will talk about that briefly again. Everything in this first 3 weeks, 2 weeks, 3 weeks is briefly. They get oxygenated, and by your muscular contraction, blood comes back to the heart. Okay, when it comes back to the heart, they come back through pulmonary veins. Pulmonary veins are the only veins in your body that has O2. This one has a lot of CO2. So it's oxygenated blood. The term they use is oxygenated blood. It comes to the right atrium. From right atrium, there is a valve right here. Guess what's its name? Left atrioventricular valve. Right atrioventricular valve, left atrioventricular valve. This is on this left side. How do you know it's left side? Imagine the heart is sitting in your body, and the way, sorry, oh, shoot, go back. Okay, so the way it's sitting in your heart is the apex of the heart is that way. So it's on my left side, and I'm looking at the left side, right side. Okay, so the apex of the heart is your left atrioventricular valve. So when somebody draws a boy, I'm horrible at drawing heart. Yeah. And draw a heart like that, that's the left ventricle right there. Okay, so from left ventricle, the blood goes from left atrium to left ventricle, very muscular. Look how muscular it is. Of course, this is septum of the heart, but look at the thickness of the muscle. Here, and look at the thickness of the muscle. Right ventricle has to pump the blood to the rest of the body. Okay, and that's why it's so muscular. It has to be thick. So the blood can get all the way to your brain, the blood can get all the way to your toes. And then of course there is a valve, so the blood does not flow back, and that's called right here, aortic semilunar valve, right there. Aortic semilunar valve. So the blood does not flow back into the left ventricle. And then the biggest blood vessel in the heart is your aorta. And there is a lot of pressure in there. I've heard stories during surgery, a surgeon accidentally cut the aorta and blood shooted all the way 9 feet. There are textbooks. I think it's 9 feet. The blood shoot up 9 feet. Okay, so you guys, you need to know 4 chambers of the heart. You need to know the 4 blood vessels of the heart. You need to know 4 valves of the heart. So 4, 4, 4, 4, 4 by 4 by 4. Look like carpentry. Yeah, sounds like carpentry. The 4 blood vessels, let's go over them quickly. I started talking about blood vessels. Superior vena cava, inferior vena cava, that's 1 blood vessel. I consider that's 1. Vena cava, okay, superior and inferior. Then you have pulmonary artery right here, goes to the lungs, that's 2. From lungs, it comes back, pulmonary vein, that's 3. And aorta, that's 4. The valves, right atrioventricular valve, left atrioventricular valve, pulmonary semilunar valve, and of course you have the aortic semilunar valve. Chambers, right atrium, right ventricle, left atrium, left ventricle. Let's go to the next slide. Okay, this is the model we have in the lab. So this is superior vena cava, inferior vena cava down here. You cannot see it very well. This is the right atrium, right ventricle. This is your atrioventricular AV valve. This is septum of the heart, and these are coronary arteries. This is your pulmonary semilunar valve, right? This is your pulmonary trunk. Well, anyhow, it's a pulmonary artery, okay? And this is aorta. This is a left atrium, left ventricle, and then of course the has been cut off. The left atrioventricular valve has been cut off, and in this picture we have to look from this end right here. I don't see the aortic semilunar valve, but in the lab, the model in the lab, you will see all of the parts. Okay. There are differences, and I hope you sit down and study those differences between artery and vein. Number one, this is the rule. All of the artery takes the blood away from the heart. Artery away. Vein brings the blood back to the heart. No exception. Arteries, in general, they all carry oxygenated blood, the blood that it went from lungs and it goes all over your body, oxygenated blood. And I will talk about that here in a minute. Hang on. And then veins, they carry deoxygenated blood throughout your body. That rule has exception. Our pulmonary artery carries deoxygenated blood. Pulmonary vein is an exception, carries oxygenated blood. Okay. So these are the differences between artery and vein. The connective tissue outside is pretty much the same outside of artery and vein. The smooth muscle in the artery is thicker than vein. And on the microscopic slides, you should be able to see that. Then the elastic fiber, elastic connective tissue is almost same, but you know, it's a little bit thicker in artery again. And endothelial in both of them is same. You have one layer of, we talked about this, endothelial cell layer. It is one layer of squamous epithelial tissue. Okay. So here are the differences. You can see the vein, since it does not have much of the smooth muscle, is collapsed. This is a vein, this is the artery, and these are nerve bundle, which we talked about it before, right? These are all nerve bundle. The most inner layer right here is endothelial. This is smooth muscle. And right here, smooth muscle. And then you have all of this from here to here is your elastic fiber connective tissue. Same as this one right here. Okay. So this area, all of that area is elastic fiber connective tissue. All of that area is elastic fiber connective tissue. All of that area is smooth muscle. All of that is smooth muscle. And then, of course, the endothelial, the most inner layer, you cannot see it very well. Okay, so another thing that artery does not have valve, but veins, they have valve. If you stand on your feet for too long, you can get varicose vein. You've heard of that one. And then so on and so forth. And here is showing you the blood goes from heart, it goes into aorta, and from aorta it goes to arteries, many of them. From arteries it goes to arteriole, from arteriole it goes to capillary, from capillary it goes to venule, and then from venule it goes into vein, and from veins it goes to inferior and superior vena cava into the heart, and then from the heart it goes to aorta. Okay. Does blood, true false question, does aorta carry blood into lungs? No, it doesn't. The only thing that carries blood into the heart is your pulmonary artery. Okay, did I talk? I've been talking. I still have 20 minutes. I can talk about respiratory system quickly and then leave the rest for later on. So here is your respiratory system. Your respiratory system, I had a professor in Kentucky. He used to say, imagine a tree and make it upside down. The tree is like this, right? It has branches and the branches have leaves here. Make the tree upside down and stuck it down your throat. That's your respiratory system. Here is the trunk of the tree, the trachea. And then it branches to bronchus. From bronchus, it branches to bronchioles. Bronchus branches to bronchioles. At the end of the bronchioles, you have these air sacs, which we call them alveoli, right here, which is called alveoli. And then these air sacs are surrounded by capillaries. This is, for example, air sacs. This hand is capillary around them. So in order to pick up the oxygen from them and give them at the same time carbon dioxide. Okay, that has a chapter by itself in your physiology class. Okay, and then of course your left lungs, they have 2 lobes because of the heart, and your right lungs have 3 lobes, and so on and so forth. Again, I'm not going to talk about all of the details, parietal, pleural, and the rest. So your bronchioles are surrounded by, just like a rope, by smooth muscles, okay? And that's why people, when they have asthma attack, these smooth muscles, for unknown reason, they contract, and that's why the person is short of breath. Okay, here is a bronchiole. This is a microscopic slide of what you had in the lab. And these are all alveoli. This is an alveoli, alveoli, alveoli. And the tissue here is simple squamous epithelial tissue. Name of structure, alveoli. Okay. So we talked about this a little bit, and then the air sacs, alveoli, and then, okay, this is important. So you should know when you, at night when you're asleep and you're not thinking, the inspiration and expiration takes place automatically. You're sitting in front of TV, you're not thinking about your breathing. The only time you're thinking about your breathing, you should be thinking about your breathing, is when you're meditating. Okay? So what happens? What happens during normal respiration and expiration? The atmospheric pressure right here is greater than the alveolar pressure, the pressure in your alveoli. So the air just forces itself in, you without even you thinking. Of course, there is in your medulla oblongata and your pons, there are the control centers, they're controlling your breathing and everything else. But most of it is pretty much going the air. The difference of the pressure, that's what this diagram is showing you. The atmospheric pressure is 760 and the alveolar pressure 758. Only 2 atmospheric pressure difference. So the air rushes in. Then the pressure in the lungs, the alveolar pressure, becomes 762. 762 push the air out. Of course, there's diaphragm here pushing it up, so the air goes back up to 760. So it's a difference of the pressures between atmospheric pressure and your alveolar pressure causes you breathe normally during just a normal respiration. Again, not doing exercise and not doing meditation times. And that's what this diagram is talking about. Okay, human pulmonary and systemic circulatory system. So the circulation between your heart and your lungs is called pulmonary circuit. Pulmonary circuit. The circulation between your heart and rest of your body is called systemic circuit. Systemic, it means all over. I'm not considering oxygenated and deoxygenated blood. But while we are here, and the circulation between the heart and your lungs is called pulmonary circuit. This is pulmonary circuit. This is systemic circuit. Okay. While we are here, let me go over this quickly. So the blood from the right side of the lungs, let's review this a little bit. It goes into, the numbers are there. Oh yeah, okay. Let's go over everything then. Okay, so what happens when the blood goes from the right side of your body, it goes to the lungs and it gives off carbon dioxide and pick up oxygen. It comes back to the heart, right? And heart pumps it out throughout the rest of the body. So the pressure, they call it partial pressure of the oxygen in the arteries is about 100. And the partial pressure of carbon dioxide in the arteries is about 40 millimeter of mercury. Okay. The other hand, in the vein, the pressure of the oxygen is low, is 40, not 100, and the pressure of carbon dioxide is 46, is higher. Okay, of carbon dioxide is higher in the vein than in the arteries. So blood comes into their lungs, and lungs pumps through aorta, pumps the blood into the tissues of our body. That tissue's body cells, it could be liver, muscle, brain cells, bone cells, you name it, kidney cells, anything in your body. So what happened, those cells, bio-1, they went through cellular respiration, and cellular respiration creates carbon dioxide. Carbon dioxide are released from the cells, and of course, for mitochondria, you remember the Krebs cycle and mitochondria electron transport chain need oxygen right here. The oxygen goes into the cell, and carbon dioxides are expelled from the cell, and they go to the vein. Of course, capillaries are surrounding the tissues of a body. This is all capillaries right here. Okay, so then the pressure of the oxygen becomes 40 from 100. Remember that? From 100 becomes 40, and then the pressure of the carbon dioxide from 40 becomes 46. I don't expect you to memorize those numbers. Do not know if it helps you to understand the concept. You're a number person. Sure, do it by all means. But you should know where the pressure of oxygen and carbon dioxide are greater and lower. Okay, I hope I'm making some sense. In the artery and veins, and so on and so forth. Here we go. He's going to some details of what's happening, carbonic anhydrase. Okay, that's physiology. I'm not going to get into it, but if you guys need to know a little bit more detail, you want to dig into it right here. That slide tells you everything. The next one is it's 49 after. Let's leave kidney for next lecture, and I think we're done. After kidney is digestive system, and after digestive system or endocrine system. Let's go over the kidney too. It's not much. It just takes a few minutes, and then now it takes 5-10 minutes, and then after that, well, what does kidney do? It filters the plasma of your blood. Liver filters the solid portion of your blood. You remember a plasma? I draw a test tube for you guys earlier. Okay, I don't have spacing here to draw it. So it filters the plasma of the blood. That's what the function of kidney is. That's it. Well, it has a few other functions, but for this class, okay, for this class, that is the function of the kidney. So anyhow, we have 2 kidneys, of course, and then 2 ureter, 1 bladder, Oh, okay. Oh, come on. 1 bladder, 2 kidneys, 2 ureters, and 1 urethra. The urethra in male is longer, so the odds of male still get urinary infection. Female urethra is shorter, so the odds of them getting urinary tract infection is more than male. The bacteria have to travel longer distance to reach the bladder. Okay, in case of kidney, and then God forbid if the bacteria even travel further and infect kidney, then that's not so good, not so pleasant. But anyhow, so that's why they say, quick lesson, wipe yourself from front to back. Do not, when you go number 2, do not wipe yourself forward, wipe yourself backward. So you take away the bacteria from your digestive tract, take it away from your urinary tract. Okay. So kidney. Kidney has 2 layers, the cortex layer right here, and the medulla layer. Medulla means middle, inside. Cortex, it means outside in biology. Cortex layer, medulla layer. And it has a capsule. Well, we will not talk about capsule. Blood from aorta comes into renal artery, and blood from kidney leaves through renal vein, and from renal vein ends up into inferior vena cava. Okay, so when the blood comes into, and I might go back and forth between these 2 slides, just hang in there. So the blood that arrives from renal artery, it branches to arterioles, right? We talked about that, just talked about that. Aorta branches to arteries, arteries branches to arterioles, arterioles branches to capillaries. And capillaries dump the blood, the wastes and carbon dioxide into the venules. Venules, they come together, dump the blood into the vein, and veins from all over the body comes and dump the blood into vena cava. You know that. That's a nice review. So when the blood, and I come back to this picture in a minute, hang on. So when the blood comes from renal artery, it goes and branches to afferent arteriole. Afferent arteriole, it means afferent arrive. And it goes through afferent arteriole and branches into capillary, which is called, those bed of capillaries called glomerulus. Bed of capillary here is called glomerulus. And from this bed of capillary, blood leaves and goes back into efferent arterioles. And from efferent arterioles, then it goes to capillary. I'm going to show you some pictures. And from that capillary, it goes into renal vein, and from renal vein into inferior vena cava. Okay. So outside of the glomerulus, this is glomerulus, a bed of capillaries. We have the name for it, it's called glomerulus. There is a structure called Bowman's capsule. Bowman's was the name of the scientist who first discovered it. Okay. And then what happens when the blood is percolating in here in the glomerulus, the red blood cells are this, and the small molecules can seep through this glomerulus right here. They can seep through and they go into Bowman's capsule. So this would be Bowman's capsule area. All of this area, I'll put a star. That would be Bowman's capsule area. And this is glomerulus area. Okay. So small molecules seep out. When the small molecule seeps out through here, there is a gap. Large molecules cannot seep out. Only small molecules seep out. When the small molecules seep out, this is not a plasma anymore. This is called filtrate. It's plasma here, right? This is red blood cells, RBC. This is plasma here, right here. All of this is plasma, but when the small molecule seeps out, it's called filtrate now. So you have filtrate here, and plasma here in the glomerulus. So the filtrate goes from, okay, let me go back and show you this picture if you don't mind. So here is afferent arteriole, blood percolates through glomerulus, small molecule seeps out to Bowman's capsule, and the blood leaves glomerulus through efferent arteriole. From efferent arteriole, blood goes around and it goes into capillaries. From capillaries, it goes into venules. From venules, it goes into veins and eventually end up into a renal vein. From venules to vein. Okay, I hope you got the big gist of it. And I will come back to collecting duct, all of that in a minute. So this diagram, if you would, this is Bowman's capsule right here, okay? The tubing that is attached to Bowman capsule is called proximal tubule. Do I have pictures anymore? No, I guess not. So this is proximal convoluted tubules. You see how convoluted they are? Okay. And these proximal convoluted tubule, they end up into loop of Henle right here. You can see that right here is loop. These are all capillaries. The red is capillaries. It goes to loop of Henle. From loop of Henle, the filtrate goes back up into distal convoluted tubules. And from distal convoluted tubules into collecting duct. Okay, I will go over that. So I talked about this, how does filtration takes place. In the microscopic slides you have in the lab, this is glomerulus, 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 and all of these are convoluted tubules. Which one is which, I don't know. I don't know if anybody can tell or not. Which one is proximal convoluted tubule and which one is distal convoluted tubules. This is the cortex layer of the kidney. The tissue here is simple squamous epithelial tissue. The tissue here in the convoluted tubule is proximal. It's a cuboidal epithelial tissue. Okay, here's a larger picture, glomerulus, convoluted tubules. If you look at these cells, it's beautiful, gorgeous looking cuboidal. And these are squamous epithelial tissue. And of course, this is Bowman's capsule right here. Here's a scanning electron micrograph of glomerulus and convoluted tubules. Okay, so let's wrap it up. Basic theory of nephron. The blood arrives through, I wrote on the pressures for you and everything else. Do not worry about it too much. But blood arrives through afferent arteriole. It percolates through glomerulus. It leaves glomerulus through efferent arteriole. From efferent arteriole goes to the capillaries, from capillaries to the venules, from venules to renal vein. What happens, there is a plasma here, but right here, the small molecule seeps out. It goes to the Bowman's capsule. The Bowman's capsule, now you have filtrate. In the Bowman's capsule, you have filtrate. So the yellow is filtrate and the red is plasma, of course, plus red blood cells. When I say plasma, it's red blood cells too. So difference of pressure causes the small molecule seeps out, and the gap is small enough so it allows the plasma to be filtered from small molecules, not large molecules. So filtrate is same as plasma plus small molecules. Of course, plasma has large molecules, filtrate doesn't, and a few other things. So the filtrate goes from here, from Bowman's capsule to proximal convoluted tubule, right? This is all proximal convoluted tubule. It goes to loop of Henle. From loop of Henle goes to distal convoluted tubule. From distal convoluted tubule, it goes to collecting duct. And from collecting duct, right here, this is collecting duct, it goes to minor calyx, from minor calyx to minor calyx, minor calyx, minor calyx, minor calyx to major calyx, from major calyx to renal pelvis, from renal pelvis to ureter. Okay, so quickly, one more time, this is the model you have in the lab. So here is a afferent arteriole. It goes through juxtaglomerular apparatus. Do not worry about that. It goes to glomerulus, blood. I'm talking about blood. And then blood from glomerulus goes to efferent arteriole. From efferent arteriole, it goes into capillaries right here. So that would be afferent arteriole, I cannot see them very well in here. It goes to capillary. From capillary, it goes into venules. From venules, it goes to renal vein. From renal vein to inferior vena cava. So the fate of plasma. Plasma from glomerulus, it goes to the small molecules go to Bowman's capsule. From Bowman's capsule to proximal convoluted tubules. From proximal convoluted tubules, this is all proximal convoluted tubules. This is glomerulus. This is Bowman's capsule, Bowman's capsule, Bowman's capsule, Bowman's capsule. Bowman's capsule covers up like that. The glomerulus. This is glomerulus. This is Bowman's capsule. Okay. So, and then the filtrate goes from Bowman's capsule to proximal convoluted tubule, from proximal convoluted tubule to loop of Henle, from loop of Henle to distal convoluted tubule, from distal convoluted tubule to collecting duct, from collecting duct to minor calyx, from minor calyx to major calyx, from major calyx to renal pelvis, from renal pelvis to ureter. Ureter to bladder, from bladder to urethra, and out through the body. Yes, okay, another thing you should know, because later on during semester we come back to this counter current multiplier. What happens during when the filtrate is, this is physiology, but I'd like to introduce you to this term, counter current multiplier. When the filtrate is moving along the nephron, the loop of Henle and distal convoluted tubule, what happens? Molecules leave and molecules come back to filtrate. So they call it counter current multiplier. Okay, it's like salt leaves and low premature ability to urea, impermeable to sodium chloride here, but water leaves, and then so on and so forth. Just know molecules, if you want to study this in detail, I don't want to spend too much time on it, come and go. Digestive system, let's leave it for next lecture. All right.